the chance to come out here. It's been a really great meeting so far, or a really great visit so far, and I'm looking forward to chatting with more folks afterwards. So what I want to talk about today is some of my work on hybridization and speciation in blue butterflies. So sort of as an overview first, it's important to recognize that variation is a ubiquitous characteristic of all life. And a central focus of my research is trying to understand the processes that affect and structure both genetic and phenotypic variation in the wild. And I sort of address that by thinking about a number of different things, from trying to sort of parse the relative effects of demographic history and selection on patterns of variation in nature. I spend a lot of time thinking about spatial and temporal variation and selection and how that can lead to adaptation and the evolution of species. And also about hybridization, both as a tool to understand the speciation process and as a potentially important component of the speciation process itself. And it's really this that I'm going to focus on today, although I'll sort of touch on some of these other topics as well. In addition to thinking about these sort of processes related to variation, I'm interested in how variation is sort of partitioned and organized at different biological levels of organization, from genes and genomes to individuals, populations, and species. Along those lines, I want to sort of start by kind of laying out two views for the organization of biological diversity. And first, I'm going to think about what I'm going to call sort of a classic view, where we think about gene flow as being a strong, cohesive force that really holds populations of a species together. So there's only limited genetic differentiation. You need really sort of strong local selection, often with trade-offs in order to really get differentiation. And where the different genes in an organism's genome function as sort of a co-adapted complex, such that there's little gene flow between species, and this sort of holds more or less across the entire genome. As an alternative view, though, it may be the case that there isn't really sufficient gene flow to actually act as a strong cohesive force in many species, and rather selection could be the primary cohesive and disruptive force. Cohesive in the sense that when populations occupy a similar environment, they'll be selected to be similar, and when they occupy dissimilar environments, then selection will act in a more disruptive manner. Sort of along those same lines, it may be that gene flow between species, or hybridization, is sufficiently common that it really can't be neglected either, and that in fact the realized effects of gene flow between species could vary across the genome because of variation in the targets of selection and in rates of recombination. And under these sort of conditions, there's a very different organization of biological diversity where you have a much fuzzier species boundaries and less clearly delimitable entities that exist. So in their sort of extreme views, probably neither of these things really capture how biological diversity is organized and works, but I think there's some utility in thinking about where individual groups of species might fall in this continuum to better understand the organization of biological diversity in a general sense. And that's sort of what I want to think about today with respect to hybridization and speciation in Lyciides butterflies, one of which is shown right here. So what I hope to convince you by the end of the talk today is, at least for these butterflies, species boundaries are indeed quite porous and fuzzy, that hybridization has been common in this group, and that it's not just some incidental thing, but seems to be associated with diversification, and that admixture followed by selection have led to sort of the repeated evolution of similar phenotypes, and to repeated parallel evolution. So to start, just sort of a little bit about these butterflies. These are Lyciides butterflies here. These are Lyciides is a whole Arctic genus. I'm going to talk mainly about only about Lyciides in North America, though. They're specialized on various legumes, primarily things like lupin and astragalus. They exist sort of as a species complex in North America. There are three recognized species: Lyciides itis, which is sort of found from throughout Canada, which I'm not showing here, and down into the Rocky Mountains some. Lyciides anna, that basically hugs the west coast of North America and gets up into British Columbia. And Lyciides melissa, that's sort of found throughout the west and farther east. They actually get out here as well. These species were originally delimited based on differences in the sort of size and shape of the male genitalia. They also differ in terms of wing pattern, the size and sort of intensity of some of these wing pattern spots. And they generally occupy different habitats and tend to specialize on different individual species of plants, although they all are sort of feeding on the same genera of plants. In addition to these sort of three named lineages, there are at least three unnamed, although I give them names here, unnamed lineages of these butterflies that are of interest to me. So all three of these things, which I'm calling the Alpine Lyciides, Warner's Lyciides, and Jackson Lyciides, are populations or groups of populations of butterflies that tend to display intermediate morphological characters between those three species I was just talking about. They tend to occupy extreme environments relative to the parental species, so relative to the named species anyway. So all three of these are found at high elevation in sort of alpine habitats, particularly Alpine and Warner's Lyciides. And as I'll sort of argue throughout this talk, all three of these things seem to be lineages of hybrid origin. So what I want to basically do today is start by describing overall patterns of spatial and taxonomic patterns of genetic variation in this species complex to sort of lay a framework for addressing two specific hypotheses about hybridization, the organization of biological diversity. The first of these hypotheses is that these montane lineages, these sort of unnamed lineages, arose through repeated hybridization events. And second hypothesis I want to think about is that selection was a key determinant in the genome composition of these admixed populations. Not by any means the only driver of genome composition, but a key determinant of genome composition. So many of the results I'll show you come from one data set, so I'll talk about that data set for just a second, and I'll point out where I'm pulling in other data sets as we go. So the main data set I'm going to talk about has samples of butterflies from 66 localities. That includes all three of those nominal species plus those putatively admixed lineages, 1,521 butterflies total. The data set is a double digest genotyping by sequencing data set. So basically where we use restriction enzymes to chop up the genome and sequence the subset of the fragments produced by doing that. For most of the results I'm going to talk about today, I didn't have a reference genome when they were done, although I do now for several of these, and I'll bring some of that in too. So this has all been sort of aligned to a pseudo-reference genome that is essentially a collection of consensus sequences of the various fragments. All told, we're looking at about 500,000 single nucleotide variants sequenced to about 7.5 coverage per individual per locus. And the vast majority of the results I'm going to show you, though, are taking into account that sort of specific sequence coverage and the sequencing errors rather than working with called genotypes, which I think is pretty important. So let's see what we have here. So the first thing I want to show you is a basic statistical summary of genetic variation across this species complex. This is a principal component analysis based on just 15,000 of those 500,000 single nucleotide variants, and these are specifically the 15,000 that are common variants that overall have a minor allele frequency of greater than 5%. So for now, I want to think about these common variants. This is an ordination of, in essence, the pairwise genetic similarities between all the pairs of individuals. So here you see the first two principal components together explain about 90% of the variation in genetic similarity across individuals. This is capturing a lot of the variation out there. Each of these little colored X's represents an individual. The numbers represent the populations that are also shown over here. And the colors are colored by the sort of nominal taxa or made-up name for them that I gave them. So the couple things I want you to see is, one, that individuals tend to cluster by population. So we see sort of overall population structure. In other words, there are a bunch of X's around the numbers. Looking across the first principal component, you'll see that it essentially separates out the two of the nominal species. So Lyciides anna, that west coast thing, is on my far left right now. And on the far right is Lyciides melissa. The other nominal species is here in the middle, Lyciides itis. And all these other things here in the middle are these putative admixed lineages. So we see that for the most part, the admixed individuals are intermediate with respect to overall sort of genome composition or genetic makeup, which would be consistent with them being of hybrid origin, but also with various patterns of isolation by distance. And I also want to point out that this generally sort of recreates the geographic pattern, but not perfectly. So for example, here you see neighboring populations of that Alpine Lyciides in blue and Lyciides melissa in green that are very, very close geographically, but separated by some distance in terms of genetic variation. Here's sort of another look at the same thing, basically. These are pairwise measures of genetic distance between all the pairs of populations, so all 66 localities, and geographic distances between them on the x-axis. The points are colored based on whether, so they're colored a color if the two populations in the pair are from the same nominal taxon, and they're colored gray if they're from different nominal taxons. And so I fit a model that has sort of whether or not they're from the same species or thing as one variable, and then the geographic distance is another variable. Both of those are important for explaining this variation. So you see in general, as geographic distance increases, you get more different populations. That makes sense. And in general, things from different species or different named entities or different unnamed but now named entities are more different from each other than things that are the same. But there's a lot of noise in there, right? So you have individual colored dots here, particularly with respect to this alpine Lyciides, that are more different from each other than are various pairs of populations that are nominally the same thing. So there is spatial and taxonomic structure, but there's also a lot of noise. It's not very clean. It's also the case that, particularly with respect to these putative admixed lineages, these alpine Lyciides, Warner's Lyciides, and Jackson Lyciides, most populations are composed of individuals that have ancestry. In other words, they've inherited parts of their genome from a mixture of different putative ancestral source populations. So what you're looking at here are estimates of admixture proportions from a model that's very similar to the model in structure, but one that I've modified to sort of account for that sequence coverage and error rates in sequencing. But other than that, it's basically doing the same thing, where each individual is a vertical bar, and they're colored based on the proportion of an individual's genome that's inferred to have come from each of these different ancestral source populations. We're looking at assuming 2 to 8 different source populations. I don't want to worry too much about that. I want you to focus on the sort of overall pattern, that particularly looking at these last 3 things, Alpine, Warner, and Jackson, most individuals have a mixture of ancestry, regardless of the K you look at, from multiple source populations. It gets less true as you get to more source populations, but for the most part, it's true. So far I've been talking all about these common variants. So with respect to common variants, we see sort of spatial and taxonomic structure. We see evidence of consistent with admixture. But what about rare variants? So it turns out that the vast majority of those 500,000 single nucleotide variants are not common but are rare. By rare, I mean they have a global frequency of less than 1%. So that makes up 70% of the total variants out there. And they have a very different spatial structure. In fact, they're very, very spatially restricted. So here's a principal component analysis based on those rare variants instead. And whereas for the common variants, the first 2 principal components explain like 90% of the variation. Here, the first 12 principal components don't even get anywhere close to that. They explain like roughly 15, maybe 20% of the variation. And rather than capturing broad geographic patterns of population structure, they're basically pulling out individual populations or a few populations relative to everything else, which is consistent with the fact that these rare variants are basically common in 1 or 2 populations and absent elsewhere. The other interesting thing about rare variants, unlike common variants, they show almost no evidence of admixture. So here's that same sort of plot of admixture proportions estimated from an equivalent number of rare variants. And you see that most individuals have bars of all one color. There's a little bit of evidence of admixture in a few populations, which incidentally happen to be where it looks like there's recent and ongoing hybridization. But generally speaking, most individuals seem to have come from a single population when you look at rare variants relative to common variants. So far, what I've shown you is that both rare and common variants exhibit spatial structure, but sort of at different spatial scales, and that common variants show evidence of admixture, whereas rare variants do not. I think this is important because it really suggests that different types of variants have experienced sort of different historical processes, which makes a lot of sense because rare variants are likely more recent variants and thus won't be affected by processes that occurred before those mutations arose. But depending on what type of variation you look at, you can get very different patterns of structuring of that variation. But from here on out, I want to focus more on common variants, and I want to focus on admixture and leave behind the basic spatial structure stuff. And as I said before, I want to address 2 specific hypotheses having set up these basic patterns. The first is that these putatively admixed or montane lineages have arose through multiple repeated hybridization events. So they've arose via hybridization, and this has happened multiple times. And then second, that selection was a key determinant of the composition of the genomes of these admixed populations. So let's think about the first hypothesis, that montane lineages arose through repeated hybridization events. These Warner's Alpine and Jackson Lyciides are the product of repeated hybridization events. So 2 predictions one can make that would support this hypothesis are, first, that the overall relationships among these taxa would not fit well to a strictly bifurcating tree. A bifurcating tree would not describe them well. And second, that if this has happened multiple times, that the genomic makeup of these different lineages should be different. Their genome composition should vary if these are multiple or repeated origins. So let's look at those predictions. So first, I fit a model with approximate Bayesian computation, really 3 models, to look at whether the relationship between each of these putatively admixed lineages is best described as bifurcating or not. So here's a sort of model where you have an ancestral population that splits into Lyciides Anna and Lyciides Melissa that then experience admixture, and you get, maybe with some ongoing gene pool for a while, and then you get these admixed populations. This would be Warner's or Alpine. And for this data set right here, this is actually a different data set. I just have data on Alpine and Warner's, not on the Jackson Lyciides. I compare that to models where the Warner's or Alpine Lyciides are sister to Anna, for example, or sister to Lyciides Melissa. Well, it turns out that the answer is that the model with the highest posterior probability is indeed the model where you have an origin with admixture. So in both cases, the model where the admixture model it has a posterior probability of greater than 50%, lower probabilities for the other models. So this is consistent with a hybrid origin of these taxa. I looked at this another way, though, using the software tree mix, which in essence builds first a maximum likelihood tree that is a bifurcating tree based on correlations in allele frequencies across many loci. Here again, we're at that 15,000 common SNP data set, and then adds migration edges to that tree to try to increase the fit of the overall tree with migration edges to the data. So here, a tree with 7 migration edges explains the data fairly well. Let me talk about this a little bit. So we have our basic tree structure here. The colors here represent different populations, colored based on their nominal taxon. And these are the migration edges that show evidence, that show instances of admixture. And their intensity is proportional to the sort of proportion of the population coming from the other population. So red ones mean that basically half this population here is made up of individuals that were from here. So a couple things I want to point out. First, here's that Alpine Lyciides group. You see multiple instances that have inferred admixture between Melissa and that group and Anna and that group that are consistent with this idea of a hybrid origin. In fact, you see some that are early on, sort of at the base of that clade, and others that may only involve subsets of those populations. So you may have had an early admixture event followed by additional instances of admixture for individual populations. You see a similar thing for Warner's Lyciides here, which is sort of actually at the base of the Alpine group here. We don't find evidence of admixture from this analysis for Jackson Lyciides. Rather, it sort of has this, it just sort of shows up as along at the base here between Melissa and Ides. If you do go to higher numbers of migration events, it does come out too, but the evidence isn't nearly as strong. So this is consistent with admixture is at the origin of Alpine Lyciides and Warner Lyciides in multiple events, some affecting subsets of populations. Now, I've also just begun to look at this at more of a whole genome level, so I now have whole genome sequences for Lyciides Melissa, Lyciides Anna, Alpine Lyciides, Warner Lyciides, and Lyciides Ides. And here I'm just looking at individual maximum likelihood trees that I've made for sort of 10 kilobase chunks of the genome. So looking at sort of different trees you get as you scan along the genome, would the expectation be that if these things are of hybrid origin, some chunks of the genome suggest that Alpine Lyciides are more closely related to Lyciides Anna, some suggest that they're more closely related to Lyciides Melissa. So these are the 6 most common trees, and together they make up about 30 or 40% of the basic tree topologies. And you see just what you'd expect, that in some cases Warner and Sierra, so here the Alpine is labeled as a Sierra, I apologize for that, but same thing. So here they're most closely related to Lyciides Anna. In other cases, they're coming out with Lyciides Melissa. So again, the genomic data seems to support the genotyping by sequence data in suggesting a hybrid origin of these lineages. So I think those data argue fairly strongly for a lack of a fit of a strictly bifurcating model. My second prediction was that the genome composition of these admixed lineages should vary from population to population or lineage to lineage. So before I hop into that, I want to talk about what I mean by genome composition. So I'm mainly thinking about things in terms of ancestry blocks. In other words, I'm thinking about chunks of the genome that are inherited from different ancestral populations, in this case, the different species. And you can think of this sort of conceptually as you start with, imagine coloring Lyciides melissas chromosomes white and Lyciides annas chromosomes blue. In an F1, you get a white and a blue chromosome. As you get additional generations of mating and recombination, you start to break down the size of those chunks due to recombination, and they can also change in frequency due to drift and selection. Now, you can sort of think about two different ways of sort of quantifying the overall composition of an individual's genome. One is in essentially what is their admixture proportion. In other words, the proportion of their genome that comes from each of those parental populations or species. So in this case, an F1 would have 50% from each species. And an F2 could have roughly the same thing, right? I mean, it could be more or less than that, but it could be in the same ballpark. Another way to think about it, which really helps distinguish between sort of early hybrids and older hybrids, is in the proportion of an individual's genome where they're homozygous for ancestry from one species, homozygous for ancestry for the other species, or heterozygous for ancestry, where they have one chunk from each of the parental species. So, here, this F1 is heterozygous for ancestry across its whole genome, whereas this individual here would only be heterozygous at this small chunk and would have bigger chunks of homozygous ancestry. So having large chunks of heterozygous ancestry is consistent with being an early generation hybrid. So you can see some of that here, that basically we're looking at a gradient here in overall in admixture proportions going from pure A to pure B. F1s are right in between and have an interpopulation heterozygosity or an interspecies heterozygosity of 1. You have back crosses basically fall on this line right here, which is the maximum interspecific heterozygosity that's possible. If you sort of have a hybrid swarm, you tend to be up in here somewhere where you have lots of segregating variation for ancestry. As a lineage gets older and older, you get more and more of these homozygous chunks. And you could stabilize anywhere in this region, but with low levels of interspecific population or interpopulation heterozygosity. So this is sort of, these two measures are how I want to think about quantifying genome composition. So what do we have? So here's the same sort of type of graph with the data for alpine Lyciides and Warner Lyciides. So alpine Lyciides here on the left. So on the x-axis, we have that admixture proportion, the overall proportion of an individual's genome that comes from one of the two species. On the y, you have the proportion of the genome where they're heterozygous. And again, back crosses would fall along this line. F1s would be there. And basically, the null expectation for a hybrid swarm would be more or less along this dotted line. So in these cases, the parental species are in orange and gray. Interestingly, we show some back crosses in those, which I wasn't expecting before doing this. I didn't really think there was that much recent hybridization, but there is some. But all the admixed individuals, the putatively admixed individuals, here the alpine ones are these black triangles. Here they are for the Warner Lyciides. Notice that they have intermediate amounts of their genome from each of the species, and they have some segregating interspecific ancestry. In other words, there are some loci where they have one allele from one population, one from another, but it's much less than you'd expect if they were F1s or back crosses or even a hybrid swarm. But these are older hybrid lineages. Their genomes have not completely stabilized, but there are many places in the genome where they have ancestry from only one of the two species. We can sort of look at that another way. So here's just a box plots looking at, this is just the first measure, the proportion of the genome from one of the two parental species. And here are the different localities for alpine Lyciides. And this is pretty cool. So first of all, you see that within a given population, so here's one population, there's almost no variation in the proportion of their genome from each of the two species. So within a population, they're all about the same. But if you look across populations moving up and down those mountain ranges, all of these alpine populations, you get whoppingly different genome compositions. You go from basically being 70% Lyciides melissa to being 10% Lyciides melissa as you move across that mountain range. So you have very different genome compositions for the different populations. You see the same thing roughly for the Warner Lyciides. There are fewer localities there. And it's a smaller range, only from about 0.5 to 0.65, which kind of neatly fits more or less within this gap for the alpine Lyciides. So if you take them all together, you basically have everything from almost pure one thing to almost pure the other thing. It seems to be more or less stabilized within a population, not completely, but sort of getting there. So I think, I hope that convinces you that these montane lineages are indeed the product of admixture, and that there's been multiple bouts of admixture giving you these different genome compositions. The next thing I want to think about is, or test really, is whether selection was a major driver of those genome compositions, or really had very little to do with those sort of what we get out the other end. Now, the first point I want to make before diving into that is that despite this, so I just showed you all this variation in genome composition for the alpine Lyciides, right? But nonetheless, they're all very similar in a number of ways. So all of the alpine Lyciides are found in an alpine habitat. This is all above tree line in the Sierra Nevada. They're all specialized on the same host plant. They all use an alpine endemic host plant of Astragalus whitneyi. They also have a number of behaviors that they share in common. So here's data from an oviposition preference experiment where we're looking at the preference of females for various host plants in terms of egg laying. And these are sort of Bayesian posterior probabilities for the probability that a female from a population will lay an egg on a given plant. And here are Astragalus whitneyi in red. So these two populations here, Carson Pass and Mount Rose, are two of our alpine populations. Note that they have an almost perfect preference for their host plant. And both of them have a very similar perfect preference, whereas everybody else is much more ambivalent about host plants. Some of them prefer their native host. Some of them prefer actually the alpine host. But overall, they're all over the place and don't have nearly as strong of a preference. Another unique aspect of the alpine populations is their actual sort of the way they lay their eggs when they lay them. So for the most part, butterflies lay eggs on host plants. They kind of glue them to the host plant. And this is a nice thing to do. These butterflies overwinter or diapause as first instar larvae inside of their eggs on the host plant. So normally, if you lay your egg on the host plant and then go to sleep for the winter, the plant senesces. These are all perennials. The next year, the new growth emerges. It emerges in the same place as the old growth, and you're right there where your egg hatches. You can eat the plant. It makes perfect sense. But up in the alpine, the same thing basically happens, but during the winter, or sort of pre-winter, as the plant material senesces, it generally gets blown off the mountaintop from the strong winds up in those sort of wind-stepped slopes. So if you stay attached to the plant, you might end up 2 miles away down the other side of the mountain. So these eggs, instead, fall right off the plants whenever they're laid. So the females lay the eggs on the plants. They're not glued to them. They just roll off and end up in the rocks next to the plants, which is, in fact, where the next year's growth is going to be. And this is, again, something that's found across these different alpine populations, despite this whopping variation in the overall proportion of their genome that's coming from these two different species. I think this in and of itself suggests that selection is at least partially responsible for how you're putting these two different genomes together, because some of the same bits are getting put together to give you these same traits across the board, right? That selection is resulting in this phenotypic similarity despite the overall differences in how much of the genomes from each of the populations or species. But another prediction that one could make, if selection is a major driver of what gets put into these genomes when they go together, is that it should be somewhat predictable. That we should be able to predict the composition of hybrid genomes, either due to processes that are operating in the parental species or based on certain aspects of the environment or ecological interactions. And that's what I want to think about now. And I'm going to sort of do a small sleight of hand at the moment, because I've been mainly focusing on alpine and Warner Lyciides, where I've done the most work on looking at evidence for admixture overall. I'm now going to switch over to talking about Jackson Lyciides, where I've done much more work in pursuing this question. I assume the same things are probably operating in both cases, but I'm talking about a different subset of populations primarily here. So the Jackson Lyciides are that third lineage where we have slightly weaker evidence of admixture from the tree mix analysis, but still lots of evidence overall. They're found basically at not quite as high of elevation as the other alpine lineages. These are more at like 7,000 to 8,000 feet, maybe up to 9,000 feet. And this is a hybridization event between Lyciides itis and Lyciides melissa. In this area, Lyciides itis is feeding mainly on Astragalus miser, which is the same host plant actually that the hybrids are feeding on here, so they don't have a host plant difference. And Lyciides melissa in this area feeds almost exclusively on alfalfa. So first point I want to make here is that in order to sort of understand how we're going to make these predictions about the genetic makeup of these hybrid populations, I want to talk a little bit more about quantifying ancestry and admixed populations and what selection and drift do to genetic variation and ancestry in hybrids. So all else being equal, divergent selection between parental populations should increase levels of genetic differentiation at those loci under selection. There are obviously many complications involved in that, and recombination rate can play a role. And there's nowhere near a perfect correspondence between those things, but on average, selection tends to increase differentiation. Likewise, selection can affect the frequency of different blocks of ancestry in hybrid populations. So again, I'm sort of thinking about things in terms of sort of painted chromosomes here. if you have a combination of painted chromosomes that decrease fitness in hybrids, then selection is either going to remove one of those two chromosomes from the admixed population, or it's going to confine them to alternative genomic backgrounds where they don't cause problems. And you can see that here's some results from simulations where I've sort of looked at this. So here on the x-axis, we're looking at chromosomes. On the y-axis, you're looking at two measures of genome composition. The black line is the overall proportion of an individual's genome from one of the two parental populations, so like your admixture proportion. The dotted line is the proportion of the genome where you're, is the proportion of individuals that are heterozygous for ancestry at that locus. And then here, these little triangles are regions under selection. And I'm looking at very many different genetic architectures here. So in all these cases, we're looking at underdominance. These last two are epistatic interactions between loci. These bottom two right-hand corners are looking at strong selection. S's of 0.65 and 0.2. These are all weaker selection. So what selection does to ancestry frequencies in admixed populations depends a lot on all these details. But I just want to point out that when you have strong enough selection, you see at the loci of selection, under selection, pretty whopping deviations in ancestry relative to other regions of the genome where there's not selection. But when you get weaker selection, this does largely go away. So what I want to talk about, so what I want to do is find a way to quantify these frequency of ancestry blocks essentially in these admixed populations. And I've developed a way of doing that using these Bayesian genomic clines, where we're basically modeling the probability that a given individual has ancestry from a specific source population at a locus as a function of a parameter that describes integration in the population at that locus and that individual's overall admixture proportion or hybrid index. So here is an example on the x-axis. I have a hybrid index going from pure species A to pure species B. And here's the probability for a given locus of having an allele from, for example, species B. So if you sort of, as a null expectation, you would just say, as the black line says here, that the probability of having ancestry at that locus from species B is simply equal to the proportion of that individual's genome that's from species B. So if you're 50% A and 50% B, at any given locus, you have a 50-50 chance of having an allele from A versus B. Selection and drift can modify that probability by increasing the frequency of one ancestry block relative to the other. And we have a parameter here, alpha, that's describing that, where positive values of alpha say, well, for this locus, there's actually a greater chance of having an allele from species B than you'd expect given your hybrid index, whereas negative values tell you you have a decreased probability of having an allele from species B relative to null expectations. The second parameter is slightly more complicated, which I've labeled beta here. Again, we have our null expectations here on the black line. With positive values of beta, you have these solid blue lines that basically are saying that, in essence, measuring the sort of admixture linkage disequilibrium between that locus and the overall genomic background, such that with positive values of beta, species A alleles are more likely more likely than null expectations to occur in a species A background, and in a species B background, they're less likely to occur. And the converse is true for negative values of beta, where there's less of an association between hybrid index or admixture proportion and the probability of ancestry, where there's essentially more free flow across the hybrid zone. So these two parameters together are capturing those two main things that I said selection would do. So if selection is purging one allele from the admixed population, then you're going to see positive or negative values of alpha. Whereas if selection is confining alleles that are interacting in a negative way to alternative genomic background, you're going to see positive values of beta. And again, negative values of beta would be more indicative of adaptive integration, where alleles are just moving freely across that zone. So use these to sort of think about quantifying patterns of integration. So the first thing I'll point out is that we see heterogeneity in genetic differentiation between Lyseides itis and Lyseides melissa. These two putative parental species differ across their genome and how much they differ. Not too surprising. So here's a distribution of FST just across loci. You see it ranges from about FST of about 0.06 to 0.3. And I have these two lines here that are somewhat arbitrary, but are basically indicating loci that have statistically exceptional patterns of differentiation, more than you'd expect sort of based on the overall distribution of differentiation across the genome. So we have variation in genetic differentiation, which could be due to genetic drift, or could be due to divergent natural selection. It's almost certainly some combination of those two things. Similarly, we see variation in patterns of integration across the genome. So here are those genomic clines for a number of loci. Here's that null expectation. The gray lines are cases where alpha and beta are not significantly different from 0. The black lines are cases where alpha is significantly positive or significantly negative. In this case, there were no significant deviations for beta. That's largely because the overall distribution of hybrid indices is very narrow, which makes detecting deviations at beta almost impossible. So we see variation in alpha with about over 3,000 of these SNPs showing significant deviations in integration relative to the genomic background. And again, this could be due to a combination of, and certainly is due to a combination of genetic drift and selection. So I have both genetic drift and selection probably driving these patterns. I want to think about how I can better parse whether it is indeed selection shaping this or not, or how much selection is affecting that. So is selection responsible for this heterogeneous integration, this sort of the specific genomic makeup that's happening in these admixed populations? And again, sort of two predictions that should hold if it's true. So first, if divergent selection is a major cause of the high genetic differentiation loci between the parental species, and the same selection pressures are operating in the admixed populations, in other words, if the same loci that are important for divergent adaptation are also under selection in hybrids, then I should see a correlation between those measures of differentiation, FST, between the parents and that measure of integration, alpha, in the hybrids. Those things that are more differentiated should have more exceptional integration. Second, so to get to the second one, recall that the hybrids are on the same host plant as Lyciides itis, which is different than the Lyciides melissa host plant. They're also in an environment that's much more like the Lyciides itis environment. So both Lyciides itis and the admixed populations here occupy sort of wet montane meadows with admixed populations tending to be a bit higher, whereas Lyciides melissa is on alfalfa in these dry fields. So if the selection that's happening is specifically tied to habitat or host plant, then we would expect that for the things that are most differentiated between melissa and itis, in other words, those things that are potentially responsible for host plant or habitat adaptation of local host plant or habitat, we would expect those things to show excess Lyciides itis ancestry in the hybrids. Right? The same alleles that are adaptive in itis should be adaptive in the hybrids. We specifically expect not just sort of an exceptional pattern of integration, but in a specific direction, with Lyciides itis alleles being favored. Let's see what we see. So here we have a plot of genetic differentiation, FST, on the x-axis, that integration measure alpha on the y-axis. Each of these little circles is a locus. The little contour plots here are showing you the density of points. Dark gray points are cases where alpha is significantly different from 0. And this red line is basically a threshold, again, a somewhat arbitrary threshold for defining things as statistically exceptional with respect to FST. The only point I really want to make here, although it may not be super obvious from here, is that there's a positive correlation between the absolute value of alpha, so high or low, and FST. It's a correlation of about 0.15. So there is an association between FST between the parents and integration in the hybrids. Cases where the parents are more different, you see more exceptional on average integration, patterns of integration in the hybrids, where alleles from one of the two parents are being purged from the population. Some of that can be explained by statistical power, but it's not the whole story, and I can talk more about that if anyone's interested. Now on to the second prediction, and that is that we should see excess probabilities of having Lyciides itis ancestry at those specific loci that are most differentiated if habitat and host plant dependent selection are a major driver of these patterns. That's exactly what we see. So for those most differentiated loci, about 90% of them have positive alpha. In other words, have a higher probability of Lyciides itis ancestry. Only 10% have a higher probability of Lyciides of melissa ancestry. And that holds if you go even to sort of more extreme outliers. And this is not just a product of overall genome composition. If you look across all markers, roughly half have alpha greater than 0 and half have alpha less than 0. This is specifically for those highly differentiated regions of the genome, which is really hard to explain by any other means than selection being partially driving that, or a major determinant of that, and it being somehow tied to things that are similar between itis and hybrid habitat or host plant. So what I hope I've shown you is that hybridization and admixture have occurred repeatedly in Lyciides butterflies. They're not something that's necessarily incidental, but rather they seem to be tied to diversification. Their exact contribution to speciation is unknown, but given the large proportion of the genome that's almost certainly involved in adaptation in any sense, there's almost no question that alleles from both species matter in adaptation. Second, I think I have evidence from sort of phenotypic data and from patterns of integration and differentiation that at least for some of these populations, selection is contributing to the overall genomic composition of these admixed populations. Selection is helping to shape and pick, in essence, the parts of the two different genomes that are going into the admixed lineages. Drift is obviously important too. And last, that despite this sort of phenotypic similarity across these admixed populations, their overall genomic composition is quite different. So we have lots of variation in overall genome composition, but not necessarily at the parts that matter, that seem to be similar across populations, even though overall they're whoppingly different beasts. So I think in general, I think this type of complexity isn't necessarily unique to Lysiades. I think you see things like this in other systems, and I think it sort of challenges our sort of more traditional notions about trying to think about the world as organized into sort of easily or semi-easily or possibly even delineated entities that are hierarchically structured in a nice, clean way. That the world could be much more complicated than that. And with that, I'll take questions. Anybody? Yeah.
For your last question, since you said it's a challenge,
Yeah.
that's really great when I was listening to this. Is it one of when that mixture occurred? You said they're ancient admixtures.
Yeah, yeah.
These big genomic differences among these alpine populations. So is it just when they were connected, or is it, you think, something else structuring the genome?
Yeah. So I mean,
Deterministic anyway.
I think it's a lot of that's the differences in overall composition. I think are probably largely demographic. I don't have any good basis for that necessarily. Maybe I have two. So one is that, again, that there seems to have been. I mean, if you trust the tree mix results, there seems to be multiple cases of admixture. Some that involve everybody, some that happened just for a subset. So the ones that happen just for a subset, you're interjecting a second dose of one species, and that's going to throw off the overall composition, right? So that's certainly part of it. And just even just from genetic drift, if you isolate, so if you imagine you have a single admixture event at one point in time, then the populations start to differentiate and are not collected by too much stream flow, then there's going to be some variation in genome composition just by genetic drift, by those ancestry blocks drifting in frequency. Subsequent admixture is going to add more to that. The other interesting thing, though, is that it does seem to be kind of clinal as you move up and down the Sierra Nevada, going from sort of mainly one species to mainly other species, which of course could be consistent with selection or genetic drift, but again suggests that there's something sort of historical processes probably played a role. Anybody else? Yeah. Yeah.
Is that it should be more similar to one parent because they have a similar
That basically, so that for the specific, so if if those things that are most differentiated between the parents are due to selection for host plant and habitat, and if the same thing is true in the hybrid zone, that the things that are under the strongest selection in the hybrid zone are associated with host plant and habitat, because Jackson Lysiades are on the same host plant and the same habitat as one of the parents, for those specific loci, they should have ancestry from that parent in the hybrid zone. Not overall, but at those specific loci we see that are most differentiated, which is exactly what we see. That where the parents differ most, you end up with Lysiades itis alleles, not Lysiades melissa alleles in the admixed population.
And are there loci that you might expect would sort of be more differentiated between those two, the hybrid and one of the parents, so that they could maybe not share so much niche overlap?
Yeah, there could, so that they're separated in space too, and I think that largely gets around that. And that's, I mean, in this system in general, geographic isolation is really important, I think, right? So you tend to have patches of host plant, and there's a population there, and the dispersal rates are really low. So you tend to get lots of local differentiation on these little patches of host plant where you have different populations doing different things. And then occasionally something happens and two populations crash into each other, admix, and more or less merge because there's not that strong a reproductive isolation overall. So without the spatial isolation, I think if you took all the Lysiades butterflies in the country and put them in in this room, it would just all collapse to one giant hybrid mess really quickly. What? Yeah.
Those individuals you sequence, did you find something like F1?
So in the very, very few. I found some that look like back crosses, mainly in the parental taxa, not in the admixed populations. The one exception is there's one population, and maybe go back to it. Actually, I can go to it there. Yeah, so so this one population on the far right side is the one case where it looks like there's actual ongoing admixture, and it's the one place where it looks like there may actually be some F1 individuals, but it's the only place I've ever found anything like that. It's an interesting case where there's lots of evidence for admixture, but there's very little evidence for current hybridization. And again, I think mainly it's spatial, that when populations there's just not much dispersal, there's not much gene flow, but there's also not much reproductive isolation.
So you're saying historically there was some contact, but now they are.
Exactly. So I think you have populations that are here and here differentiating, then they get a period of contact, and they probably just merge more or less at that point. But then there are other populations that didn't do the merging trick, right? So you have this sort of mosaic of differentiating populations and periods of admixture between individual populations, not so much this sort of steady state of like some gene flow back and forth. That's not really what's happening here.
So could you estimate when the hybridization or?
I've tried that a couple of times, and I'm going to keep trying it. I never have got numbers I'm truly happy with. There's so much uncertainty in these sort of historical analyses, right? So I've done some sort of coalescent estimates of the admixture times, and I guess they're reasonable. And sometimes, so the answer is it's sometime in the last 1,000,000 years, and they come in with that, but they're very broad. They don't really narrow it much. They don't narrow it down much more than I knew before I did the analysis. With the Jackson ones, it's got to be even more recent because where all the hybrids are was glaciated until about 14,000 years ago. So it probably happened as butterflies poured into that valley and mountain area after glaciation. That one's probably more recent. I'm doing stuff now looking at actual sizes of ancestry blocks in these hybrids to try to get at another way of getting at sort of the age of the admixed populations. But I don't have more than 10,000 years, less than 1,000,000 years is sort of where I'm at right now that I can confidently say is true. Yeah.
Do any of those highly differentiated SNPs occur in characterized genes?
Yeah. So I've looked at that a little bit. So most of these markers are not in genes from the get-go, and there's really not enough that are highly differentiated and in genes to make much of a compelling argument for it. I've looked at it in terms of, so I have other work I've looked specifically at mapping performance on different host plants, and there I've done a little bit more digging in on that, where you get more sort of SNPs of interest, and there too you don't get an enrichment for genes. Period. You can get enrichments for certain functions, but only if you sort of cheat, because it turns out that you sometimes have multiple SNPs that are near each other that are all basically tagging the same gene. So if you count them all, then you get an enrichment. If not, then it's like everything shows up once or twice. The only thing that really comes out kind of cleanly is that you do, for host plant adaptation, for basically SNPs associated with performance, are overrepresented in transposable elements, which is kind of interesting. But that's all I know from the functional standpoint. I mean, I think particularly for something like host plant adaptation, it's such a multifaceted thing, right? It's not one thing. There are nutritional differences among plants. There are phytochemical differences. There are structural differences. There's behavioral differences in terms of oviposition. There's phenological differences. You're probably talking about hundreds, if not thousands, of genetic variants that are contributing to host plant variation in various ways, or in host plant adaptation in various ways, such that you're not necessarily going to find one thing that's really sort of driving it. Cool. Thanks.